In this video I'm going to talk about autosomal linkage. For the AQA specification, we've got need to show genetic diagrams to interpret or predict the results of crosses involving sex linkage, autosomal linkage, multiple alleles and epistasis. So let's take a look at this autosomal linkage. Before we start, just remind you that dihybrid crosses, I'm going to draw a little cell here, dihybrid crosses involve the interaction of genes on different homologous chromosome pairs. So if we have big R and little r of a heterozygous individual, and this is homologous pair 1, and we may have some kind of texture gene if we were talking about the P's, and this would be a homologous pair 2, so 1 and 2. This interaction leads to the 4 gametes, which eventually leads to the 9:3 to 3:1 ratio of a homologous or heterozygous cross of dihybrid individuals. So what is happening with autosomal linkage? The autosomes are in humans the 22 chromosomes other than the sex chromosomes, and when we're talking about linkage, if we have one homologous pair, so I'm going to label that as 1 in there, and we've got big R and little r. We also have big G, little g on the same chromosome, so they are linked because they're on the chromosome. This one may be from mother, this one may be from father, or male, female. So if we then take these during meiosis, and the DNA duplicates, we end up with big R and big R, big G on our chromatids, which will eventually lead to a gamete with big R, big G, and another gamete with big R, big G. And then this chromosome also duplicates. So we have little r, and little g on these chromatids, which eventually become little r, little g gamete, and little r, little g gamete. Now, that means there's only 2 different gametes possible, but thinking about dihybrid, we end up with big G, big R, little g, little r, little g, big R, and big G, little r, which results in far more combinations and therefore variation. So let's take these letters I've used, or these alleles, and actually use them. So I'll take it to P's. We've played with P's before, so I'm just going to do R equals round, so dominant R for round, and we'll have little r equals wrinkled, so recessive, so just making this up. Big G is green, which is dominant to little g, yellow. So that's what we're going to look for in our phenotypes. So let's say we've got 2 P's that are heterozygous, so we're going to go with a heterozygous round green P with a heterozygous round green P. P's. Genotypes. So we're going to have heterozygous for round and heterozygous for the green coloring. And we'll cross that with heterozygous. So what do our alleles look like? Based on what we've just done up here, we're going to end up with big R, big G, because they're linked on the same chromosome, and little r, little g, and likewise here These are our only options. So I'm going to add sex in here. I want to do my Punnett square. Let me move this up a little bit. So if you're doing this with animals as opposed to plants, we have male and female. So whatever our gametes are. So now that we've recognized or identified our gametes, so we get big R, big G, r, little g, big R, big G, r, little g. And we cross those over, what do we get? So big R, big R, big G, big G, big R, big G, little g, big R, big G, little g, r, little g, little g. So, final F1 offspring genotypes: big R, big G, big G, big R, big G, little g, and there's 2 of those. Just going to write them all out. And finally, r, little g, little g. Phenotypes then. So if we come back to look at our dominant versus recessive, we have round and green, round and green. Spell green properly. There's the dominance, round and green again. And finally the 2 recessive together, so we get wrinkled and yellow. Ratio of phenotypes then, 3 to 1, so we've got 3 round and green, to 1 wrinkled and yellow. Now, we could also go on to talk about crossing over and the variation that introduces, but for right now, let's stop right here.